Our names are Karen Delgado and Abigail Hale, and this presentation is on the results of our lab research project, the effect of liquid fertilizer concentration on the lifespan of C. elegans.
The purpose of our experiment was to study the effect of fertilizers on human health. Our proposed hypothesis was that different levels of fertilizer will have an effect on the lifespan of C. elegans. When designing this project, we decided to use the nematode C. elegans because it was easy to keep alive. The necessary environment is easy to replicate and sustain with agar, E. coli, and temperature regulation. The nematodes are also easily accessible to us as high school students and economical. We found it necessary to find an organism that was relatively inexpensive to obtain and sustain since our school did not have a lot of funding resources. C. elegans are also commonly found in nature, so we knew that C. elegans are an organism that could be exposed to the environment which we were creating.
C. elegans were also an advantageous organism to test on, as they have highly similar systems to the human, including nervous, digestive, and reproductive systems. C. elegans also highly conserve human genes, making our results highly applicable to human health. Collecting data on the C. elegans was very easy, and we only needed a dissecting microscope. This data could be collected in the limited time span of one class period, creating another reason why it was a good organism to use. When trying to decide a fertilizer to use, we selected a liquid fertilizer because of limitations with our equipment, which included not being able to fully dissolve the solid fertilizers unto the point where we could manipulate them to produce the necessary concentrations. The first step in our procedure was creating enough agar to pour the appropriate amount of plates to last us the entire experiment.
The following step in our experiment was to calculate the levels and concentrations of fertilizer to be added into the agar. We used the recommended 2 times and 3 times the recommended amount for our concentrations. We also cultured E. coli to inoculate the plates with the day after. We allowed the plates to grow overnight, then transferred a single C. elegans egg from an unused plate onto the new test plate.
After the egg hatched, grew into an adult, and laid its own eggs, we transferred the original worm onto a new plate as eggs were laid, in order to keep the worm isolated and easily accessible. The data was collected on the worm while we were moving it in between plates, using a worm pick and a dissecting microscope. The positive and negative controls for our experiment were acquired from a previous experiment. The negative control was held on normal Diffco agar and allowed the worms to live approximately 2 weeks. Our positive control was held on Diffco agar with salt added, which caused the worms to die within 24 hours.
After collecting our data, we graphed our results using a Kaplan-Meier curve. We received a p-value of 0.2718, which is far greater than the p-value of 0.05, which is the required value for a statistically significant experiment. Therefore, we did not have enough evidence to reject the null hypothesis, which states that fertilizer affects the lifespan of C. elegans.
Using 30 worms for the entire experiment, we did not generate enough data from our sample sizes for each concentration to prove any kind of link mathematically between the lifespan and use of fertilizers on C. elegans. Our recommendation would be to use at least 30 worms for each individual concentration to generate more data.
The low, medium, and high data appeared like a bell-shaped curve with survival rates of 30, 70, and 50% respectively. This may be due to the different levels of nitrogen and phosphorus concentrations within the fertilizer, with low providing not enough nitrogen and phosphorus to the worms, and high possibly providing an overdose. This would support why the medium had the highest survival probability out of all 3 concentrations.
We also noticed irregularities in the movement of our C. elegans. We noticed that by the end of the experiment, the C. elegans had little to no movement, possibly suggesting other health effects. The worms in our experiment also had very hesitant movement, as they were possibly searching for uncontaminated areas. C. elegans have sensors along their body for environmental factors, which could be an explanation for this. The worms in our experiment also circled, a technique for removing substances from their body, much more than worms on normal agar. One possible solution for this would be that something in the fertilizer was irritating the worms. These questions would need to be answered in detail before a link between lifespan and fertilizer could be supported. Until further studies are completed, we must state that our results do not support a strong connection between the lifespan of C. elegans and the use of liquid fertilizers.